everyone. We're going to talk a little bit about our blood. And as a system, it's not just the blood that we're going to be talking about. We're going to be mostly talking about how the blood gets around the body, and then what is the purpose of having blood and the importance of having blood. One of the things that you might think we'll be talking about today is blood and clotting. That actually I'm going to to that in a different section, mostly when we talk about the immune system, which is coming up soon. So anyway, the blood system itself, we're going to mostly talk about blood, veins, arteries, and then the heart is going to be the main focus of the blood system. So the central organ, of course, for pumping the blood throughout the heart, throughout the pumping the blood throughout the rest of your body is your heart. But that seems obvious to us now. That actually was not something that was always known to be, and it wasn't until I guess you can say relatively recently that we looked at the heart being the organ that pumped the blood around. You're going to read an article soon that's going to talk about that discovery and how it kind of contradicted some previous thoughts that we had about the heart and its role, as well as the blood and how the blood got around. And we're going to use that as a way to talk about theories in science and how theories get modified based on new evidence. So anyway, there are 4 chambers to our heart that you should be familiar with, 2 atria and 2 ventricles. There are 4 valves then that would connect each of these chambers to another chamber. 2 are semilunar and 2 are atrioventricular. If you took anatomy, you might have learned these as different terms, and I can't really tell you the history behind who uses which terms and why, but in our context, we are going to be using the terms semilunar and AV, or atrioventricular valves. We will not be using things like mitral valve, and so that might be challenging for those of you that did take anatomy, but as you see a lot of diagrams and working some stuff on your own, it should become a little bit easier for you to go back and forth between those. 5 major blood vessels that we're going to be focusing on that are extending from or into the heart: the aorta, the superior and the inferior vena cava, and then the pulmonary artery and vein. And one of the main things that we want you to be able to do is identify all aspects of the heart anatomy. And soon we're going to be doing a dissection lab, which will allow you to explore that in a lot more detail, very hands-on, and then you will be able to use that information to try to create some sort of mechanism for yourself to remember these different components and structures of the heart. So when we look at an outline or diagram of the heart itself, these are the things that I would expect you to be able to do. And so if you looked at these without using the numbers here, or the names here associated with the numbers, I would want you to be able to identify all the main structures that are in here. And so the superior vena cava, the inferior vena cava, superior meaning on top of, inferior meaning below. The aorta is a pretty easy one to identify based on its size and its location. Pulmonary artery and vein, we will explore those in great detail when we get to the dissection. These are going to be a little bit harder for you to identify in terms of looking at them in the actual heart, but the coronary arteries are things that we're going to talk about in terms of consequences for damage to them. Sorry for shifting over too soon, but if we shift this over and look at some of the inside views, right atrium, left ventricle, left atrium, left ventricle, these are things I would expect you guys to be able to know. And then the different valves, tricuspid and mitral is what you guys probably talked about in anatomy, but what we're going to talk about again is the idea of what the atrioventricular valve is, which are the ones that connect the atrium and a ventricle, and then we're going to talk about the semilunar valves, which are the valves that are bringing back into or leaving the heart. So knowing knowing the chambers, knowing the different valves, these are all going to be important things that you're going to have to be able to do and possibly draw on an IV exam. So when we look at 15, 16, and 17, again, like they're listed here, so instead of the tricuspid valve, it's going to be the right atrioventricular. Instead of 16 being the mitral valve, which is right here, that we're going to look at as the left atrioventricular. And then 17 and 18, 18 is not shown, but we'd be able to utilize these as the right semilunar and the left semilunar. So you'll get used to these. Don't worry about the differences here yet. So the blood flows to the heart in the same path over and over again. And not only knowing the anatomy, but knowing that pathway is something that we want you to be able to be really familiar with. And one of the key things to understand is the difference in terms of blood circulation. So there are 2 ways that blood gets circulated in our body. And we're going to talk about those different systems, and you need to make sure that you can keep track of what each of those systems are. But this is the main diagram that you should be really familiar familiar with in terms of blood within the heart and how it's moving. And so you're going to go from right atrium, you're going to go to right ventricle. From right ventricle, it's going to leave, and it's going to go to your lungs. When it leaves and goes to your lungs, it's going to come back from your lungs and enter the left atrium from the left pulmonary vein. From the left atrium, it's going to enter the left ventricle, and then from the left ventricle, it's going to enter the aorta, and then from the aorta, it's going to be distributed throughout the rest of the body. And if we look at the difference in the left and the right ventricle, one thing should be very apparent to you here in this diagram, and it's going to be very apparent to you when you look at them in the heart dissection lab that we're doing. But if you look at the ventricular walls here, the right ventricle has relatively thin walls of muscle, but the left ventricle is very thick. So I want you to think about why that would be. What would be the biggest difference in terms of why we need thicker walls or more muscle in the left ventricle as compared to the right ventricle? And we'll explore that when we do our dissection in a lot more detail. The two distinct paths I was referring to are the pulmonary circulation and systemic circulation. So keep in mind that the lungs have their own distinct pathway for blood. And that's obviously very important because we know that the blood is what's going to be transporting both carbon dioxide and oxygen. And so as we get oxygen in from our lungs, we're going to need it to go back to the heart so it can get distributed throughout the rest of the body. And as the heart collects the blood that is lacking in oxygen and high in carbon dioxide, that needs to go to the lungs to get rid of the carbon dioxide. And so keep in mind, and please be very aware of the two different circulation systems that we have. One is lung-based only, the other one is going to be essentially anywhere but the lungs. How to remember what is what? So when we have all these different parts, are there some tricks that we can use to remember what's an atria, what's a ventricle, and the like? So I look at a couple of different things. So arteries. When we talk about the difference between arteries and veins, there is a misconception that I hear quite frequently that, well, veins carry deoxygenated blood. Arteries and veins have nothing to do with the amount of oxygen that's in the blood. Arteries are always carrying things away from the heart, and veins are always carrying things to the heart. And so the blood that leaves your heart and goes to your lungs, for example, it leaves and does that through the pulmonary artery, but it has no oxygen in there yet. It's not until it picks up the oxygen from the lungs and carries it back into the heart through the pulmonary vein that it's oxygenated. But then from the pulmonary vein, it will get distributed with oxygen, lots of oxygen in it, to the rest of the body through arteries that way, mostly the aorta. And then what about atria and ventricle? Well, if you want to try to keep those straight, just if you picture a traditional kind of form of a heart, and the bottom of that heart kind of forms a V, we can think of the fact that if you take the heart and cut it in half this way, that you'll have the atria on top and the ventricles on the bottom. Just a little trick here to help you kind of remember those and keep those straight. So after leaving the left ventricle, the blood's going to enter the largest artery of the body, which is the aorta. From the diagram before, and you can kind of see up here, it's large, it branches out the top. There's a huge aortic arch, and then it bends around and curls behind the back of the heart, and then that will extend all the way down through your abdomen, and then it will split and distribute blood into each of your legs. There are a lot of branches along the aorta, and one of the first leads to the coronary arteries, and those coronary arteries are the ones that will supply oxygen and nutrients specifically to the heart. So the heart is a muscle, and just like any other muscle in your body, it's going to need oxygen to be able to function. If you get a blocked coronary artery, that's one of the main causes of what is known as a heart attack, and there's lots of things that we can consider a heart attack, but a blocked coronary artery is one thing that can lead to that. And a heart attack, in its simplest form, is essentially the heart either stopping to pump or pumping in a completely irregular rhythm, and a blocked coronary artery can cause that to happen. You will actually be doing some research, and we'll discuss that at the end here, about the consequences and causes of coronary arteries. So if we look at what a blocked coronary artery would actually look like, so it's relatively small. So if we look at the aorta here, it's a very, very large blood vessel. And this first branch right here is going to lead to the coronary artery. And if you zoom in on the coronary artery, if you get a buildup of these arterial plaques, it will physically constrict the volume of blood that can flow through here. And then you're going to get blood clots that can possibly develop, or you're going to get a slow flow of blood that goes through here. And if the tissue that is surrounding this does not get the amount of oxygen that it needs, that tissue can die. And if that tissue dies, then that's a big, big problem for the heart, because we don't need any muscle tissue in the heart dying. Otherwise, the muscle is not going to work properly. And if this muscle is not working properly, then you get big trouble throughout the rest of the body. So the contractions itself that causes the heart to beat is what we looked at previously when we did our lab on EKG sensors and how they actually work and what information we got from those was all based off these electrical impulses. And whenever the heart is beating, it's a very controlled system. It's all regulated by the nervous system, something we call myogenic muscle contraction. And the beating itself is controlled in these set rhythms. And if it wasn't controlled by this nervous portion of our nervous system, then those rhythms could break down. And if those rhythms break down, then you're going to get uneven distribution of blood throughout the body. If you have uneven distribution of blood throughout the body, then you get a big problem with the amount of oxygen that is needed in different portions of your body. We need to have a pretty consistent rhythm so that the blood is flowing in a very regular pattern to both remove the CO2 as well as to provide oxygen to cells as they need it. The right atrium is where you're going to find what we call the SA node, or the sinoatrial node. And this is the main location of the electrical control of the heart beating. So it literally acts as the natural pacemaker. And if you know anyone that has a pacemaker, what it does is it provides electrical signals to the heart to allow for that first set of responses to occur, or the first set of stimuli to occur that will cause the heart to actually beat for the first set of beats. And then that will carry over to the second set. So we always hear 2 beats kind of at once, and we saw that with the peaks in our EKG sensor, and we're going to talk about that here in a second. So that signal from the SA node is going to cause the atria, both atria, to contract. So the top portion of your heart is going to squeeze. It's going to pump blood into the ventricles, and a split second later, we're going to get a message that's sent to the atrioventricular node. The AV node is delayed by about 1/10 of a second, and we can actually go back and look at some of your data to see how much yours is delayed within the EKG. But the second delay, the 1/10 of a second delay here, is to allow its own signal from the AV node to be sent, which causes the ventricles to both contract. And when the ventricles both contract, we're going to get blood that then is sent out of the heart. And one, it's going to go to the lungs, and then the other, in the left ventricle, it's going to go to the rest of the body. Both atria contract, then both ventricles. And we hear that lub-dub, lub-dub, lub-dub of a heartbeat because you get the atria followed by the ventricles that are contracting, and then that cycle repeats over and over again. As you exercise, you might have noticed that your heart starts to beat faster. Mostly that's because we have an increased need for oxygen. The adrenaline that's in your body from your adrenal glands is going to cause the heart to beat faster. That is usually stimulated by an increase in CO2 in the blood. As you get an increase in CO2 in the blood, you get a decrease in pH. It gets more acidic as carbonic acid is produced. That is going to signal the medulla in the brain to relay a signal to the SA node from the cranial nerve that's going to cause the heart to start beating faster. And it's beating faster so that it can circulate blood more efficiently and more quickly. And by circulating more quickly, it's going to allow more CO2 to get out of your lungs so that we can provide new, fresh oxygen coming in to keep up with the demand that your cells are going to have for that increase in oxygen as they're respiring more, as you're exercising and consuming more and more energy. Blood itself is the medium that our transport mechanism within our body relies on. We need things to be in the blood to be carried from place to place within the body. We already talked about digestion, so if you think about the things that we are absorbing, how do they get to that point where they get assimilated into new molecules? They need to be moved around our body mostly through the blood. Blood's got 4 main components that allow this to happen. Plasma, red blood cells, which are also called erythrocytes, white blood cells, which are also called leukocytes, and then platelets. The main substrates that are transported within the blood are going to be nutrients, oxygen, CO2, hormones, antibodies, urea, and thermal energy. And if you want to look at some more detail in terms of how each of these things and the structure of them from what we talked about last year, how their individual structures are related to how they get transported and what is doing the transporting, go ahead and check out page 66 in your book, which is very, very early stuff that we covered from last year when we talked about different molecules, mostly macromolecules. The arteries and the capillaries and the veins are all very different structurally, and they are different based on what they're needed for. And they are the veins, the arteries, the capillaries, again, are basically kind of like the piping or the plumbing that allows the blood to get throughout the body, both away from and back to the heart. But they have different structures to them that allow them to move the blood in different ways. Arteries have a very smooth muscle layer, which obviously we are not in control of as smooth muscle, which allows for the contraction of arteries. So arteries can actually get bigger or smaller, and as they get bigger or smaller, it's going to allow us to regulate blood pressure. So the pressure in your arteries is going to be very important for that blood to be able to get around the entire body. And if for some reason the pressure is too low, then blood's not going to travel as fast as it needs to. If it gets too high, it's not going to travel as easily as it needs to, and both of those things can cause problems. But arteries are able to contract and relax to allow for a different diameter of that tube, which will then allow blood to flow through differently. And as it goes through an artery, it's going to get smaller and smaller and smaller until we go to an arteriole, which then leads to a capillary bed, which then leads to a single capillary. Single capillaries then are going to allow the diffusion of particles within our blood to get out and into the blood so that we can transport these materials back and forth as needed. Capillaries have extremely thin walls. Typically they're 1 cell thick to allow for that easy chemical exchange. That diffusion process through that semi-permeable membrane of those cells makes it really easy to do in a capillary as compared to an artery, which is very thick, has muscle around it. The exchange that would have to take place there would be near impossible. The capillary beds themselves are all going to empty into what we call a venule, and then we're going to get bigger as we go back. So venules will lead to veins, and then veins will eventually lead back to the heart, and then the cycle starts all over again. Veins have really, really thin walls, but a larger diameter. And they have little valves that are stationed kind of throughout different lengths or different portions of the veins that allow the blood to move, and it prevents backflow. So as the blood is moving through the arteries and being squeezed in there by the heart, the blood that's in the veins is kind of naturally kind of get pushed through. But in order to prevent backflow, we have these valves all throughout our venous system that allows the blood to kind of move forward and not go back until eventually it gets back into our heart. If we look at the structure of arteries and veins, you should be able to identify a cross-section as being one of these two things, and also a capillary. So if you see a very, very thin one, these are usually the easiest to identify. 1 cell thick, really, really small tube. These are going to be our capillaries. This is what a capillary bed would look like, and so as the blood flows through here, it would kind of dump into the capillary bed. Things get exchanged back and forth. They get picked up by the venules, then go back into the veins. If we looked at the veins, they have connective tissue immediately around the inside, but then they do have some elastic fibers and a little bit of smooth muscle. We're not showing you any of these valves in this cross-section, but if you look here, this is showing you the valve that are in the veins that allow them to either open or close. That prevents that backflow. So as blood pushes this way, the valve will open up, and then as it closes, the blood cannot go back down. But then if you look at an artery, it's quite different. The level of muscle within the artery is significantly greater than the level of muscle that we see in the vein. And that is usually very apparent if we look at a cross-section, which you see here of some tissue that shows both a vein and an artery. This purplish stuff is the muscle tissue, and you can see the thickness here is way thicker than the thickness here. So if I were to show you this picture, I would want you to be able to identify which one of these is an artery and which one is a vein, and be able to explain how you know that. This is clearly the one that you would identify as an artery because of the thickness of the muscle tissue, compared to this one, which has significantly thinner muscle tissue. Here's another look at the same type of thing. The adipose tissue here, that's just fat tissue that's surrounding the small artery that you see here compared to the small vein. These are blood cells that you can see within them, but again, the thickness of that tissue, and the darker the purple is the muscle, the thickness of that tissue is significantly greater than the thickness of that same tissue type in the vein here. Another thing that's interesting in this picture to note is that they do point out these capillaries, and if you look at the diameter of the capillary, you can see how much smaller. They're calling these small arteries. You can see significantly the difference between the diameter of the capillary here and the diameter of the artery here. Capillaries are super teeny tiny. And I don't have a good picture to show you for this one, but oftentimes capillaries, as you get down to the very end of them, will only be able to transport 1 red blood cell at a time across the width of the actual capillary. They don't get much thicker than that. This is a capillary, and as you see, this picture doesn't show just the one, but because it's a little bit thicker, this would be kind of higher up the chain, if you will. But I just thought this was a cool picture to show you the individual red blood cells that are traveling through the actual capillary itself, and seeing how they actually, they physically travel through it too. It's kind of like little individual subway cars going through a subway station. This is another, I thought, cool picture showing, I don't know if this one is artery or vein. If I had to judge based off of the thickness here, I'd say probably vein. But these are all actual individual red blood cells that are flowing through that blood vessel. And just the level of detail of this picture, this is a real picture. So an electron micrograph has been falsely colored to show you the differences, but you can clearly see the individual red blood cells here, and then the diameter of the blood vessel itself. If we look at a coronary artery, we can see what a healthy coronary artery looks like as compared to an unhealthy one. And so if you look at the top picture here, this is a healthy coronary artery where you have very little plaque buildup. You have wide open space, a lot of area in here for the blood to flow. But as you look and see, this is a picture further down the tube. So this is a partially blocked artery here. And from the segment that they took away from the actual aorta, this is 5 centimeters away from the aorta. You can see a little bit of blockage that's starting to build up, but for the most part, there's still a decent amount of space here. As you go further down that artery, so this is 2 centimeters further away from the aorta, you see a significantly greater blockage. And so this wall of plaque is getting significantly bigger, which is going to constrict the amount of blood that can flow through here even more. And so as the blood gets into a more and more narrow channel, it's going to have a much more difficult time to pass through. And this is where you get clogs that can build up. And if that completely closes, it can kill the tissue that's around it. Even in this case, some of the tissue that's surrounding this is not going to get nearly the amount of oxygen that it needs, which can also cause damage to the tissue. This is another cross-section of a coronary artery that has been significantly occluded. If you look at this kind of orangish picture right here, this is where the muscle tissue stops, and everything within that is all clogged. So this is all plaque that's been built up. And one thing that I'm going to have you guys do, if you look down here, I would like you guys to start working on your own, and we're going to discuss this in class at a later date. But I want you to look at the causes of this. What actually causes this plaque buildup? What are some things that you can do both from a diet standpoint, from a health standpoint, that would cause these plaques to build up? And then what is the consequence? So just because we have these clogged arteries, blood can still flow through here. What is the ultimate consequence that can happen? How does that consequence occur? Is it always the same? Is it always just a heart attack? Or are there other conditions that can be caused as a result of an occluded coronary artery, with the word occluded simply meaning blocked and not clear. So that's all I had for you today. And if you guys have questions about this, again, this is simply a recap for the most part of the material that comes from Chapter 6, and this is Section 2. So if you have questions, please bring those with you. If we can get clarifications and we can talk about those in class, that would be great. And hopefully you can take this information and start thinking about the different things you're going to look at when we see our heart dissection here in a couple days.